then preparation of injection of extraction of pituitary gland, then dosage, method of injection, and HEPA. Induced breeding in fishes, that induced breeding in fishes, which is also called as artificial breeding by hyphophysation. Induced breeding in fishes is also called as artificial breeding by hyphophysation. Hyphophysation technique, with the help of hyphophysation technique, the breeding which takes place induced in the fishes, that is called as the artificial breeding by hyphophysation. That is, we know that the major calves, basically the major calves breeds in the river throughout the monsoon, that is in the in particular June to August period. But here a technique is used by which the fishes are artificially bred. So artificial breeding by hyphophysation or induced breeding is, in that particular technique, some knowledge about location of pituitary and its dissection, preservation and storage, preparation of dosage, and method of injections is very important. So here are some points about the artificial breeding technique or induced breeding technique. So first of all, we must know the knowledge about the pituitary location. So pituitary is situated on ventral side of the brain, near the optic chiasma. Pituitary is situated on the ventral side of the brain, that is near the optic chiasma, just below the hypothalamus. This is a particular location of the pituitary gland which is situated in the brain region where near the optic chiasma, just below the hypothalamus. It contains a particular type of hormones like FSH and LH, which induce the breeding in the fish. So then second point, source of pituitary gland. So the collection of this gland is made only from fully ripe and, that is, matured fishes or gravid fish. The collection of this pituitary gland is collected. This pituitary gland is collected from the fully ripe or matured, that is a gravid fish, and the suitable period for the collection of the gland for major carp is from May to July. The suitable period is from May to July, when majority of fishes reach its advanced stage of maturity. So the suitable period is May to July. In that particular period, the fish reaches in its advanced stage of maturity, and this gland should be collected from living or just killed specimens. Gland should be collected from living or just killed specimens. Next step in the induced breeding, that is the removal of gland. So anyone can remove that pituitary gland by dissecting the head skull. The skull of the head, by removing the skull of the head, we can dissect out the pituitary gland, and this pituitary gland, when the gland is exposed, it is carefully picked up by forceps. So second important thing, after the exposure of the removed pituitary gland, we can carefully pick it up by forceps. Care should be taken so that pituitary gland is not damaged. In the next step, that is preservation and storage of pituitary gland. Then after removing of the pituitary gland, next stage, that is the preservation and storage of pituitary gland. This gland is stored in 100% alcohol. So this gland is stored in 100% alcohol in a refrigerator at ordinary temperature. Then extract of this gland may also be preserved in glycerin. The extract of this gland is preserved in glycerin, and whole gland is preserved in 100% alcohol. Then weight of the gland is found in different weight of the gland is found in different species. So in case of major carp, the weight of the gland is between 7 to 18.8 mg. This is the weight of the pituitary gland in case of major carps. The next step, preparation of injection. Next step is the preparation of preparation of gland suspension for the injection. So after removing this gland, after storage of the preservation, then this gland is taken out on filter paper. First of all, this gland is taken out on the filter paper, and then it left for short while to allow the alcohol to evaporate, and then homogenized with distilled water or 0.3% saline water. So the tissue is homogenized with distilled water or 0.3% saline. The homogenized gland are then diluted with the same liquid. The dilution is ordinarily made at the rate of 0.22 cc per kilogram of body weight of breeders. First of all, this tissue is homogenized with distilled water and again diluted with the same liquid depending upon the body weight of the breeders. Then the gland suspension is centrifuged. This suspension is centrifuged about 1000 rpm for 5 minutes. Next step is the centrifugation of that suspension at a rate about 1000 rpm for 5 minutes. Then the supernatant taken out, to make the dose. Then the supernatant fluid is diluted to desired dosage, which is drawn up for injection. In the next step, that is the dosage. So for the breeders, there will be 2 injections are given. First injection, single injection or first injection of 5 to 10 mg of pituitary gland per kilogram body weight of female, and 2 to 3 mg of gland per kilogram body weight of male breeders gives the satisfactory result. The first injection of 5 to 10 mg of gland per kilogram body weight of female and 2 to 3 milligram of gland per kilogram body weight of male can be given. So in the second injection, if first injection does not work, then the second injection is given, and which is given after 8 to 10 hours. After 8 to 10 hours, second injection gives to that breeders. Then in the next step, which is the method of injection. So intramuscular injection on the caudal peduncle or shoulder region near the base of the dorsal fin with 2 ml of syringe is given. So this injection is given intramuscularly on caudal peduncle or shoulder region near the base of the dorsal fin. And for that process 2 ml syringe is used particularly. Again, some clinical needle. The needle which is used for this injection is 24, needle number 24, needle number 22, needle and 19 are used for breeders over 1 to 3 kg and above 3 kg. In the next step, then after the breeding, these eggs are the breeders are then live in a particular happa. That happa is called as breeding happa. The breeders are then live in a particular happa called as breeding happa. The breeding happa is a rectangular box shaped box shaped container made up of mesh and made up of mosquito net cloth, and are closed on all sides to prevent breeders from escaping out. So there is having particular rectangular structure, box shaped structure, which is called as breeding heppa, and in that breeding heppa these breeders are then lived for the breeding purpose, for the spawning purpose. The breeding heppas are of variable sizes, different sizes of breeding heppas used for the different type of breeders. Then after the completion of the fertilization, how we can identify the eggs? So it is suspended inside that breeding heppas are suspended in the water. Afterward, we can identify the fertilized eggs. So the fertilized eggs appear transparent pearl-like. Fertilized eggs appear transparent pearl-like, whereas unfertilized eggs are whitish in color. So by which we can identify the fertilized eggs. And lastly, the fecundity rate of Indian major carp is about 2 lakh per kilogram body weight. The fecundity rate for Indian major carp is about 2 lakh per kilogram body weight. This is about the technique which is called as induced breeding technique in fish breeding.